we have, so here in figure 18.3, we have the TRP operon, which is consisting of the promoter and the genes of the operon. The genes of the operon code for enzymes that would connect together to form tryptophan. So this promoter consists of the promoter and the regulatory gene TRPNPR, which codes for protein that is the inactive repressor. So since the inactive repressor is not active currently, the RNA polymerase would be allowed to freely code for tryptophan. So once tryptophan is present throughout the body, then tryptophan would end up binding to the repressor, and this active repressor would bind to the operator, and which would block out the RNA polymerase. So because the RNA polymerase is blocked off, no RNA could be made, and since this RNA cannot be made, then no tryptophan would be produced. So here in figure 17.12, we have a pre-mRNA, which has a cap at the 5' end to prevent degradation from that end. So this pre-mRNA is composed of an exon, intron, and exon. But before it exits the nucleus through the nuclear pores, it must go through a spliceosome. And this spliceosome is going to consist of protein, snRNA, and other proteins. So the pre-mRNA enters the spliceosome, where the snRNA binds to the ends of the exon, the borderline between the exons and the introns, and separates the intron. So this cut out intron is then exits the spliceosome and is then degraded. So now we only have the exons left. So with these remaining exons, the other proteins, they come together to stitch the exons back together, and then this finished mRNA exits the spliceosome, and now we have an mRNA spliceosome component in that cut out intron. So once the mRNA exits the spliceosome, a poly A tail attaches to the 3' end of this mRNA to prevent degradation from that side, and once that happens, the mRNA leaves the nucleus through the nuclear pores. Okay, so here in figure 18.10, we will go over the process that takes place before RNA synthesis. Here, the activators bind. So on the DNA, activators bind to the distal control elements, which are the enhancer. So this causes the DNA bending protein to bend the DNA so that the activators would have a better shape. So at this point, we have several core proteins that then bind to activators. So first, the mediator protein binds to the activators. Then the general transcription factor binds to the mediator protein. Then the RNA polymerase II binds to the general transcription factors, binds to general transcription factors in order to, and on the Tata box, just ignore this. So on the Tata box, on the promoter. So this forms an active transcription initiation complex, and once this is formed, the RNA synthesis begins. I feel like I messed up. So here in figure 18.5, when we have the lac operon DNA. So firstly, assuming that lactose is always present in this example, but we will first talk about when glucose is scarce and CMP level is high. So firstly, because lactose is present, this allolactose has bound to the lac repressor, forcing it to become inactive. So while this is still inactive, since the end there's high levels of CMP will bind to the inactive CMP, forcing it to become active. Once it is active, this will bind to the CMP binding site and assist RNA polymerase to move along the DNA to code for lactose. However, when CMP levels are low and glucose is high, there will not be any CN, there won't be any CAMP to bind to CAP. So this will usually be inactive. However, even if the CAP is inactive, there's still small amounts of lactose being produced. Okay, so here for figure 18.4, we are going over the lactose operon portion of the DNA. So here, unlike the tryptophan operon, the repressor is usually active. So here, LAC-I codes for the active repressor that's usually blocking the operator. And since this operator is usually blocked, RNA polymerase is unable to code for lactose. However, when there's a high presence of lactose within the cell, allolactose will bind to the repressor, and this would force the repressor to become inactive. And since this is inactive, RNA polymerase can start coding for different amino acids. So it's LAC-Z coding for beta-galactosidase, which breaks down lactose into glucose and galactose, and coding by LAC-Y for permease, which allows lactose to pass through the cell membrane, and RNA polymerase coding for LAC-A, which codes for transaxolase, whose function is currently unknown. So for figure 18.3, we have DNA and the TRP operon. This TRP operon is always active until it's interrupted. So here, the RNA polymerase moves along the DNA and along genes and proteins, so this will code for different enzymes. Here they would code for enzymes C, D, C, B, and A. And once all these enzymes are coded and produced, then these enzymes will come together to form tryptophan. And farther back from the DNA, there's a promoter and a regulatory gene, TRPR. This TRPR codes for a protein that serves the inactive repressor, that serves for the repressor of the TRP operon. So once there's too much tryptophan or a high presence of tryptophan present throughout the cell, the tryptophan will bind to the repressor, and this repressor suddenly becomes active. The active repressor will then bind to the operator and block RNA polymerase's path, so it cannot code for any of the enzymes to form tryptophan. So no RNA is made, and because of that, no tryptophan is made. This whole process is called negative feedback.